and share. Okay, so I'm going to add a little layer on top of everything I've said so far. We're talking about, we have seen what light is, we have seen how we can, what happens when the light finds bits of our sample, and also we spoke about how we can manipulate light in the microscope. Now we're talking about the image formation. So I remind you what I said just before, a fluorophore is actually a light source, and it emits light all over in all directions, so in 3 dimensions. It's a very, very tiny light source, so it's a point light source. And then some of this light, only some of it, will make it towards the objective, and the rest is lost. And the amount that gets into the objective is, of course, limited by the size of the objective and the front length of the objective. So I remind you that what happens when light encounters an edge is that the wavefront is bent. And this was the whole train that I was showing you. So this is our little point source. The wavefront is changed, and we get now parallel, so a flat wavefront, and then we can do that again, and we have a intermediate image, and then we end up with the final image which we record. But depending on where we put the iris, it's called a field stop or an aperture. And what happens in here is that there is no image here. So we are in a case of aperture, where we have the edges of the objective act as an aperture. So the inner edge of the objective is an obstacle. So there is diffraction there. And it's an iris outside the image plane, so it acts as an aperture. And it influences the interference pattern, which is, again, what we are recording as an image. It affects the whole image, the quality of the whole image. It doesn't work like a carton, it affects the quality of the whole image. And so we have more lenses that are transmitting the light all the way to the final image plane, and this is what we record. At the image planes, here and here, the diffracted light waves come together from many directions, so they interfere in 3 dimensions. They bump into each other and interfere. And so we have a very complex 3D interference pattern in here that is formed, and in here as well. And this is where we put our sensor in the final image plane, and we record this 3D interference pattern that is formed by the diffracted light, and this is how we get our image. So here we have a very tiny fluorophore, it's a tiny point source, but the image that we are recording is not a point. It is a complex 3D interference pattern that doesn't have the same shape at all as our little fluorophore. So what does it actually look like, this image? What does it look like if it's not the same? So in the 1800s, there was this guy called Thomas Young, and at that time, people were probably had lots of time on their hands, and they were pretty clever. So this guy was an expert in medicine and in sound and music, and on the side, he was also studying the wave nature of light. This type of people. So anyway, this is one of his drawings, and this is the drawing that he made of the interference pattern, and I will show you. So here we have some light coming in, and you see we have an obstacle with 2 slits. If we have only 1 slit, this is what happens to the light. And if we had only this slit, this is what will happen to the light. But if we have 2 of them, like this, what happens? There is an interference pattern that forms behind the obstacles. And this is what it looks like. And you can see here these lines of the crests. This is what he drew here. So if you squint a little bit, if you close your eyes a tiny bit, you probably will see these lines here. I remove them so that you can see. Yes, so we have all this interference pattern. This guy was pretty clever, and he actually figured out that this is what was happening out on the other side of the obstacle. So we have constructive and destructive interference behind. And this is why some waves are bigger and some waves are smaller. And that forms this 3D interference pattern. Well, he was talking about 2D interference, but we know that for us it's in 3 dimensions of space because the fluorophore sends light everywhere in all directions. So he was talking about a 2D interference pattern with very tiny apertures. These apertures are so small that they are actually smaller than the wavelength of light. This is what happens then. So we can see the bright areas, they are constructive interferences, and here we have destructive interference. So it's black. If we now put a sensor here to look at what that looks like, well, this is what we can image then. So we have an interference pattern that looks like this if we have a 2-dimensional interference pattern. We know very well that our sample emits light in 3 dimensions. So our interference pattern here is 3-dimensional. And also we know that the size of the objective is much, much, much larger than the wavelengths of light. And there's only one aperture. So what happens then? Well, there was this Huygens guy, and it was even earlier. And this guy, he was a mathematician, a physicist, an engineer, an astronomer, and an inventor. Whatnot, you know? And he actually described that the case that we have is that we have a light behaves like lots of small wavelets. So we have a lot of wavelets of lights here at the aperture. And this is the reason why in 3 dimensions we have this very complex interference pattern behind. So if we put our sensor here, what happens then? What do we see? So I remind you that this would represent basically the edge of the objective, and this would represent the final image plane where we image. So there's a great video that if you want to watch in iBiology.org, and in here I have the link because iBiology, they have a lot of different courses and videos, but this is the one I'm talking about point spread function, where Jeff Lichtman, with a fantastic name to working with microscopy, he describes in a very, very nice way how the interference pattern is formed due to these wavelet things that were described by Huygens. So we have a 3D interference pattern that is formed because we have the edges of the objective. So if we have like 2 wavelets only, this is what we're recording when we put our sensor here. That's what we would record. If we put 3 wavelets, this is what we get. And you see we have a beautiful interference pattern. If we put even more wavelets, then you see that it becomes really complex. And now we get this interference pattern when we measure here. And even more wavelets we end up in here. And what you're seeing is that the light, the more wavelets we have, and the more the light converges towards one big blob like this. And this is actually what we are imaging when we image the image of our microscope. So the light coming from our sample diffracts at the inner edges of the objective, and many wavelets are formed. And so we get a super complex interference pattern like this, formed at the image plane. And therefore, the image of a fluorophore looks like this. It's elongated with a very bright center and some sort of shadows around, right? This shape is called the point spread function. And during this course, we're going to talk about the point spread function, or PSF, a lot. So now you know that the point spread function is actually the image that we are recording when we image one fluorophore. So the point spread function, or PSF, is the 3D interference pattern that is formed by light that comes from only one point light source, so one fluorophore, if you have only one fluorophore, and that diffracts at the inner edge of the objective the interference pattern that we obtain and that we image. This is the point spread function. So here we have our tiny spherical fluorophore. So it has a very nice spherical shape. And when this light goes through the objective and the microscope, if we put a sensor there, this is what we're seeing. We have a bright center and lots of circle, like rings around it. And if now we do a Z stack through our fluorophore, so we add one Z dimension and we look at the image from the side, we look at the Z stack. So this is the Z dimension and this is X. This is what we're seeing. So we're seeing exactly what Jeff Lichtman described in his video. We have a bright elongated center and a lot of patterns around it. And if you look carefully, you can see that it looks like a rugby ball. So the 3D interference pattern that we are imaging is actually like a rugby ball. And if you look carefully, you can see the rugby guy, right? If you squint a little bit, I'm sure you can see, right? Okay, maybe it's all in my head, but I think you can see it as well. So we have lots of little rugby men. Every time we have a fluorophore in our sample, what we're imaging is a rugby man. And both the excitation light and the emission light, because they both go through the objective and the macroscope, they both have a point spread function shape. And the light cannot be focused to a smaller point than this. So the point spread function is the limit of resolution. Okay? The smaller the point spread function, the better the resolution. But if we have a big point spread function, then we don't have such a good resolution. So sometimes we can image. So this is a little movie that I took of beads, fluorescent beads. So very small fluorescent beads. They are 170 nanometers in diameter. So they are below the resolution of the macroscope. So it's a point light source, and I imaged them with a wide field system. So we'll talk about what is a wide field compared to a confocal later. But the simplest microscope you can get. And I took a Z stack through the bead, and it was a 4-micron Z stack. So you see, my bead is 170 nanometers, so it's a very small bead, but I took a very, very big Z stack compared to the bead, right? So I have 41 images, and the step from one Z plane to another is 100 nanometers. So this Z step is 24 times larger than the bead. So maybe you would explain that when I'm out of focus, I don't see the bead, and when I'm out of focus on the other side, I don't see the bead. But this is not what happens. So here we have 4 beads on this image, and they're all in the same plane. And what I'm going to do is I'm going to do the Z stack, and to make sure that you see something, I've adjusted the brightness and contrast for each image. So even if it's out of focus and it's dim, you actually see what's going on in the image. And there we are, we're out of focus, and we can anyway see the beads, right? Now we are in the focal plane, and now we're going, and did you see the rings? Yeah, let's play that again. So we are out of focus, we see a blur, then we go to the focal plane, then we go out of focus and we see the ring. So the point spread function is something that you can actually see in the macroscope, because this is what we are imaging, okay? That's what we're imaging, and this is the shape of the image that our microscope creates. A very tiny light source. So the beads are visible throughout the whole 4-micron Z stack, even though the Z stack is so much larger. So we see the beads well before we are in focus, and that's because the light is completely spread throughout a big volume. So we have first a small bright dot when we are in focus, but we have some big blobs or some rings when we are out of focus. Okay, so we can see the PSF shape. So diffracted light is spread in XY, so it's also larger in XY than the bead, but it's also in Z, it's much, much larger than the bead. And that is called the point spread function. So the light that is coming from every single fluorophore in the sample looks like that. And it is diffracted to a very large 3-dimensional point spread function. So our fluorophores are very tiny spots that emit light in all directions, and the light travels throughout in 3D, but only some of it makes it to the objective, and it diffracts here at the edge of the objective, and then through all sorts of lenses inside, there's lots of lenses inside the microscope. Every time we have diffraction, we have interference, And at the end, we get a complex 3D interference pattern at the image plane. This is the point spread function. So point spread function is the shape of the pattern that we're imaging. And it is, then we put a sensor here, and this is what we are recording. So when we take Z-stack through a tiny bead, that's what we see in one image when we are at the focus plane. And when we do a Z-stack and we look at the Z-stack from the side, this is what we see. Okay, that's a summary of everything I've said before. If we now draw a line through these, you will often see these representations of the point spread function. This is a line through the XY image that is in focus. So you can see these little waves here are the rings, and then the peak at the center, which has a certain width. And then if we do the same thing through a Z-stack, you can do like a two-dimensional image like this that shows you these rings and then the center blob. Okay, so these are different representations of the point spread function that you can find somewhere. So why do we care about all this? As we know, the inner edge of the objective acts as an aperture, and apertures affect the whole image, not just the periphery of the image. So the aperture will affect the brightness of the image. So the image is dimmer if we have a smaller aperture. And also it affects how much information is in the image, and that is the optical resolution. So the aperture of the objective is called the numerical aperture. It's abbreviated as NA. This is one very important specification of the objective, and we will talk about that in the objective lecture. So the numerical aperture, and if you have a high NA objective, high numerical aperture objective, we have more lights that participate to this 3D interference pattern, which is the point spread function. And therefore, we have more information about our sample and the image is brighter, right? So high NA aperture objective means a brighter image and a higher resolution. Higher optical resolution and brighter image when we have a high NA objective. So now that I have said this, I think we have a better idea of what light is and how we can manipulate it, and also how the image that we are recording is formed. So now I want to put again this key concept of light microscopy that I spoke about before, just so that you understand maybe what I meant. The image of a fluorescent object is the result of refraction of the emitted light, the light that comes from our sample, that goes through lots of media with different refractive indices. That can be our sample, which is heterogeneous. It can be the cover slip, glass cover slip. It is also the immersion medium and the lenses. All this will affect the image of our fluorescent object through refraction. But it also depends, the image of our object also depends on how much light finds its way into the objective. So it depends on the numerical aperture of the objective. And also it depends on the way light diffracts at the edges of the inner edge of the objective. And that is also the numerical aperture. So this one is how much light, so that's the brightness, and also the way it diffracts. And so it depends on the 3D interference pattern that we record at the image plane. So this means that without diffraction and interference, there is no image. Okay, what we are recording is diffraction and interference. Without that, we don't have an image. That's it. So we have nothing to image. So therefore, anything that affects the interference pattern, so the point spread function, for example, refraction, will affect the image. That's where your role comes in. You would like to avoid that you affect the point spread function. This is the reason why I said that the image of an object is never and cannot be like the object, because what we're imaging is the point spread function. We're imaging the way the light interacts with the sample and the microscope elements. So it cannot be the same. Therefore, for you, preparing a good microscopy sample is mostly about minimizing refraction. And that will have an effect both on the excitation and the emission lights. And you do not want your sample to act like a kaleidoscope, basically. So this is why we talk about refraction index a lot, because it has an effect on the point spread function, and therefore it has an effect on your image, okay? So this is what is important when you prepare a good microscopy sample. We will see what a good microscopy sample is later. So the optical resolution that delivers light is limited by the light wavelength, and it's also limited by the numerical aperture of the objective and also the refractive index of the immersion medium. So these are the things that will affect the optical resolution. And then we have an extra layer, which we will talk not next week, but the week after, which is the digital resolution. And the digital resolution is the way we actually record the image. So that is the pixel size. So that also adds to our resolution in the image. So the bad news for you is that to fully describe a sample with very fine details, for example, if you look at vesicles, whatever sample you have, it will have edges, right? You have the edge of the nucleus, you have the edge of the cell. All these edges are fine details. So all samples have very, very fine details. And to fully describe such a sample, we would require infinite resolution. That's not good because we do not have infinite resolution. So it is not possible, that's what I said, it cannot be like the object. It is not possible to have an image that fully represents the sample. But the good news is that most scientific questions do not require you to fully describe the sample. And most of the time, you do not even need the maximum resolution of the imaging system. So you can see in here the very great importance of how you formulate your scientific question within the limits of microscopy, which is that we will never be able to describe fully the sample. It will never be exactly the same. So please design your sample to refract as little as possible. What does that mean? Place your sample on the cover slip if you can, not on the slide. If you cannot put it on the slide, for example, because you are boiling your sample for oxygen retrieval, then use the minimum amount of mounting medium, okay? And then match the refractive indices of the objective and the sample and the mounting medium, all this to prevent more refractions, prevent the sample from looking like a kaleidoscope. And again, we will talk about this later in the course. You will only notice the difference between a good and a bad sample if you compare them side by side. So if I see your image and I say your image would be better if you had your sample on the cover slip, you're not going to believe me. But if I show you, then you will see it. So this is the same sample. It's the same objective, the same settings, acquisition settings, same microscope, everything. And in both cases, we were acquiring at the best focus possible. And what happens? Do you think we have the same resolution in this image and in that image? Which one do you think is easier to segment? Because in this one, I can assure you we're not out of focus here. This is the best focus we can get. So this is what happens when we design a sample that creates refraction. But if you have this, you cannot really guess how much better it would be if you had your sample on the cover slip. So this one is on the slide and this one is on the cover slip. Okay, so this is easier to segment. We have more stable intensity, so less fluctuation, and we have a higher sample-to-sample reproducibility with this. Okay. So improve contrast. If you improve the contrast here, you actually improve the usable resolution in this case. So it's easier to segment. So keep in mind also the limitations of a microscope. We cannot have infinite resolution, and you need to have your scientific question designed for this. If your scientific question requires you to work at the limits of the resolution of the microscope, you should change technique or you should change scientific question. So working at the limit of the resolution of the microscope is extremely, extremely risky. So this is my opinion. Okay. And I just want to say that there's a very, very nice demonstration of all this stuff that I've been telling you about diffraction and interference in this little video that you're welcome to watch later. So I would like to know if there are any reflections on this, what I've been telling you so far.
I've got a question.
Yes.
So you said to use as little mounting medium as possible. How do you know kind of the compensation between using as little as possible and then avoiding that the sample dries out?
Yes, absolutely. We need to have enough mounting medium to cover the sample completely. But what we do not want is that the mounting medium lifts the cover slip away from the sample. So we would like to have just enough mounting medium so that the cover slip is against the sample. Right? We do not want to have like a big sandwich of a big layer of mounting medium between the objective and the sample. That's what we want to avoid, yeah? So I know that we've tried a lot at our facility to ask people to put their sections, tissue section, on this cover slip instead of the slide. And we'll talk about all this later in the sample preparation lecture and the day. But when we have antigen retrieval, it's difficult because it detaches, unfortunately. So then if you cannot avoid putting, and this is the reason why when you use like monolayers, it's very easy because you just seed them on glass. You can seed them, you should seed them on the cover slip, not on the glass slide. I think there was someone who was seeding their cells on the glass slide. It's much better to seed them on the cover slip. And then it means that when you're imaging, the mounting medium is not between the cover slip and the sample. But if you can't avoid it, just put as little as possible. So what you can do, and Gabriela will explain all this to you, but you can do a test and put, okay, what if I put 3 microliters? What if I put 4 microliters? You can do a test before you actually mount your sample. And then you will know exactly how many microliters you need to use.
Thank you.
You're welcome.
I also have a question. You mentioned that to reduce the refraction in the sample as much as possible, one can match the refractive index of the objective sample and mounting medium. How do we know the refractive index of the sample?
Yes, that's a good question because it's mixed, right? So as you have seen, the light, when the light refracts, it's going to change direction, right? So if it changes direction for a very short time before it's imaged, then it's okay. You don't see so much effect. But if it travels like this for a very long time, then the effect is very big. So this is why we want to reduce the amount of mounting medium. It's the same thing in a sample. If you have a thin sample, it will have a little effect on how much the light travels. But if you have a thick sample, then the effect is very big. And this is one when you have a little bit thick. And when I say thick, people who image a monolayer of cell, they know that when you image the bottom of the cell, which is against the cover slip, or you image the top of the cell, you already see the effect of refraction. The top of the cell is blurrier than the bottom of the cell. It's always like this. And that is because of the refraction index mismatch. So if we have a sample that is even a little bit thicker, we're even talking about 10, 15 nanometers, microns, sorry, or like 20 macrons. When it's like this, then you need to do something which is called optical clearing, which we will talk about, and homogenize the refractive index. So you do some chemical treatment to make the refractive index of the sample homogeneous to avoid. Yeah. So this is the way, but that we will talk about again later during the sample preparation day, how we can do that. Any more questions?
I have one question. So if I am using the water lens,
Yes.
should I submerge my sample also in PBS but
Yes,
not
absolutely. So water objectives are excellent samples to image live samples because so it depends again, of course, on the cells that you're imaging. If you're imaging cells that have a lot of fat, for example, adipocytes, then you're a little bit more in trouble. But very often, if we image cells that do not have a lot of fat, so the cytoplasm has the same refractive index a little bit higher, but not very much higher than water. And then your cells are live, so you put them in a culture medium. And this has more or less the same refractive index also as water. And so in that case, the best objective for live samples is, because of that, is a water immersion sample. Yeah.
Okay, thank you.
Yeah. Anyone else?
Can I ask a question too, please?
Yes.
Yes. So, how much mounting medium should one usually add for it to be enough? Because what we usually do in the lab is to add maybe more mounting media than we need so that it covers the whole cover slip and also the whole sample to make sure
Exactly.
that there are no bubbles in between
Yeah.
the slide and the cover slip.
So I
So
think Gabby will explain all this to you during the sample preparation day, but all these sort of things, they do make a difference. They absolutely do make a difference. So it is important to put as little as possible and not put a big blob, and certainly make no bubbles. So you will hear all this in the sample prep day. Yeah, and we have like even little videos and stuff for you to look at, even to practice.
great. Thank you.
Yes. Good. So if you guys have no more questions, we're going to go now to the workshop Zoom to have a little quiz. Okay. See you guys there, and then we'll be back here at 11:25.